everyone. I'm Etchi. I'm from UC Berkeley. I'm a PhD student in the Vision Science program, and I'm very excited to share some of my research with you guys, which is looking at the impact of autism spectrum disorder on functional retinal development. So autism spectrum disorder is a neurodevelopmental disorder which is categorized by impaired social communication, repetitive and restrictive behaviors, as well as impaired sensory processing. And another phenotype that we see in many individuals with ASD are a number of visual deficits. And one example of the many visual deficits correlated with ASD is a condition called nystagmus, which I'm showing right here, which is the involuntary movement of the eyes. And children with ASD are about 5 times more likely than typically developing children to have this condition. Most cases of ASD are idiopathic, meaning that the cause is unknown, but some cases have been linked to specific genetic changes, and many of these genes have been associated with the miswiring of neural circuits. So visual processing begins in the eyes, and our eyes are essentially a biological camera which captures light and converts light into a neural signal which can then be further processed in the brain. And for my research, I'm really interested in a particular tissue in the eye called the retina, which is this yellow piece right here. This is a light-sensitive tissue in the eye. And if we were to zoom in on the retina, we see that the retina is a complex circuit made up of many synaptically connected neurons. And it's here in the retina where all this neural processing occurs. So I'm showing you the schematic of the retina right here, and light first enters through the retinal ganglion cell layer, but it's actually first detected in the photoreceptors, which are comprised of your rods and cones. And this signal is then transmitted through the various different interneurons in the retina, which are all synaptically connected, and a synapse is just a connection between neurons. And once this signal reaches the retinal ganglion cells, the axons of the ganglion cells actually form the optic nerve, which is that connection between your eyes and your brain. So in addition to the broad visual deficits that we see in ASD, a number of studies have also found that specifically retinal function is altered as well. So a previous study has found that retinal function is altered in humans with ASD, particularly Fragile X syndrome. And what this study did, they performed an electroretinogram, which is a measure of retinal function in response to different light stimuli. And this test will give you 2 different readouts. You'll get an A wave, which correlates to photoreceptor function, and you also get a B wave, which correlates to inner retinal function. And this study found that the A wave in patients with Fragile X syndrome was not significantly altered compared to the control group. However, when they looked at the B wave, they found that the B wave amplitudes were significantly decreased in patients with Fragile X syndrome, which is suggesting that there is some deficit in the interneurons in the retina of patients with Fragile X syndrome. So next, what I'm showing you here is that in development, this is really when all of our synapses are starting to mature, and particularly in autism, all these different synapses start to be affected. So I'm really interested in looking at how retinal development could be potentially altered in autism. But during this time in development, the synapses between the photoreceptors and the retinal ganglion cells are immature. And during this time, at least in mice, the eyes are closed and the mice cannot see because there's really no way of getting the signal which is detected in the photoreceptors to the retinal ganglion cells, which are the output of the retina. And however, during this time, although mice cannot see and the circuit is still immature, the retina itself is not silent. And during this time, the retina is spontaneously generating activity, which is called retinal waves. And next, what I want to show you guys is a timeline of retinal circuit development. And retinal waves in particular last from a long period of development, from a couple of days before birth to about 2 weeks after when mice open their eyes. And what I'm showing you here is this timeline of retinal waves. So the first stage of retinal waves, which I'm not showing you here, are actually prenatal in mice, which I will not be looking at for my research. But the next stage are mediated by acetylcholine, which is a neurotransmitter. And this particular stage of waves occur from around P1 to around P9. And once this circuit is refined, the waves then transition from being mediated by acetylcholine to glutamate, which is another type of neurotransmitter. And these waves occur from around P10 to around P14, and they conclude when eyes open. So next, what I'm showing you here is an example of what these waves actually look like. And what we see here in the purple is the actual retinal tissue. And we see in green, these are these neurons that have been labeled with the calcium dye. And we see that cholinergic waves and glutamatergic waves have different spatial temporal properties. So we see that cholinergic waves, they tend to propagate in a large sweeping motion across the retina. And we like to call these waves on the beach because they kind of look like waves on the beach. And next, we also have glutamatergic waves, and we see that these waves propagate in smaller regions across the retina, and they're more like ripples. So we also know that retinal waves are essential for visual circuit refinement because in a mouse model where cholinergic waves are perturbed, we see an interesting phenotype. In particular, we see that horizontal direction selectivity, or the ability to detect motion in the horizontal direction, is perturbed. So what I'm showing you here is some data from our lab where we're looking at these eye tracking responses in a wild-type mouse and a mouse with perturbed waves. So we see in this wild-type mouse that's tracking a horizontal stimulus, we see that there is some sort of eye tracking going on, and this is what we expect to see in a normal animal. However, when we show that same stimulus in a mouse with perturbed retinal waves, this is not moving right now, but we see that the animal is actually unable to track that stimuli. And this is really interesting because in humans with nystagmus, they also have a very similar phenotype, and they lose this optokinetic reflex. So a big question I want to answer with my research is, are retinal waves altered in the FMR1 knockout and SHANK3 knockout models of autism? So I'll be using 2 different mouse models to study ASD. The first one is the FMR1 knockout, and the next one is the SHANK3. And we're really interested in these models because they both have phenotypes that are similar to humans, and they're also both loss of function models. Also, they have deficits in their glutamatergic synapses, and both of these genes are expressed in the retina. So the FMR1 gene encodes for Fragile X messenger ribonucleoprotein, FMRP, and FMRP forms complexes with other proteins, and it aids in the transport and translation of mRNAs. And mutations in this gene can lead to a condition called Fragile X syndrome, which is the most common cause of monogenic ASD. And I'm showing you an example of a normal X chromosome and an example of a Fragile X chromosome. And we see that in the Fragile X chromosome, we have these fragile sites, which is where the mutation occurs. And this is how this disorder gets its name. An absence of FMRP can lead to exaggerated glutamatergic signaling. And it's hypothesized that this is the reason for many of the neurodevelopmental phenotypes in ASD. And the SHANK3 gene encodes for scaffolding proteins at excitatory synapses. And I'm showing you an example of SHANK in the postsynaptic density. And we see that along with other scaffolding proteins like HOMER, SAPAP, and PSD95, they form complexes that help to anchor neurotransmitter receptors at the nerve cell surface. And similarly to FMR1, absence of SHANK3 can lead to another type of neurodevelopmental disorder called Phelan-McDermott syndrome, which is another leading cause of monogenic ASD. So for a quick workflow at a glance, I'll be using 2-photon calcium imaging to image retinal waves. And first, I'll be using my different groups, and then I'll be isolating the retina. And when I initially dissect the retina, it's circular, but I flat mount it into this petal shape. And then I'll be bolus loading a calcium dye, which serves as a proxy for neuronal activity. And then I will be using our 2-photon calcium imaging rig to image this tissue ex vivo. So I'm really interested in knowing if the circuits that are mediating waves are altered in our ASD models, and the way I'll be doing this is by utilizing pharmacology. And specifically, I'll be using a nicotinic acetylcholine receptor antagonist called DH-beta-E, and this will block the nicotinic acetylcholine receptors on the starburst amacrine cells and retinal ganglion cells. So during the time of cholinergic waves, there will still be acetylcholine, but they will be unable to bind to the receptors, and waves will go away. And just showing an example of what this looks like, I'm showing you some traces of before I use DH-beta-E and after I use DH-beta-E. And we see that before DH-beta-E, we have these peaks which correlate to wave activity. But then after DH-beta-E, we see that the wave activity is abolished, and this is confirming that the circuit was mediated by acetylcholine. So I really wanted to see if the frequency of waves were altered in the FMR1 knockout. and we found that when we look at the frequency in this particular knockout to wild type, there was no change, which is expected due to what we've seen in literature. And what I'm showing you here is an actual video from what it looks like when I'm imaging these waves with the 2-photon calcium imaging rig. And we see that these waves are just kind of flowing across the retina, almost like these flashes. And they're almost like these waves on the beach, essentially. And next, now that I know that retinal frequency is unchanged, I wanted to see if these circuits are still mediated by acetylcholine in the FMR1 knockout. So what I did, I measured retinal wave frequency in a group without any drug administered, and then I also looked at the frequency after drug has been introduced. And we see that in wild type, after I've used drug, we see that there is an abolishment of waves. It goes from about 1 wave per minute to no waves. And we also see something very similar in the FMR1 knockout, which is showing that in the FMR1 knockout, they're still being mediated by acetylcholine. So for a brief summary, retinal circuits are impacted in ASD, and we can use our 2 different ASD mouse models for studying the development of different synapses, like glutamatergic synapses. And retinal waves are a great assay of synapse development. And the cholinergic retinal waves are normal in the FMR1 knockout, especially with their frequency and the circuits that are mediating them. And for future directions, I plan on continuing these experiments in animals that are at the glutamatergic waving time point, and also the SHANK3 mutant animals, which our lab has just acquired. And with that, I want to give a big thank you to my lab, especially a postdoc in our lab, Dr. Rachna Devan-Samaya, who I've been working very closely with throughout this project. And I also want to acknowledge my funding source for funding this project, and thank you guys for your time and attention.